Good morning or good afternoon, depending upon where you are in the world. It's very pleasing for me. My name is Pat Blackall. I'm a Principal Research Fellow within the Centre for Animal Sciences based at ESP, and it's a real pleasure and a privilege to be the chair of today's seminar. My first role, and an important role, is acknowledgement of country. I acknowledge the traditional owners and their custodianship of the lands on which we meet. We pay our respect to their ancestors and their descendants who continue cultural and spiritual connections to country. We recognise their valuable contributions to Australian and global society. This seminar will run till 1 o'clock. At the actual end of Arla's talk, we'll hold a Q&A session. If you have questions that you'd like answered, please use the Q&A tab at the bottom, not the chat button. We won't be monitoring the chat button. So we'll address all the questions at the end of the seminar. So our seminar today is to be given by Professor Arla Tabor, who's a professorial research fellow at CAS. She leads the Animal Biotechnology group at the centre, which she's done since 2010. Arla's a true UQ person. She did her undergraduate degree here at the Department of Microbiology, then spent a year up north at JCU, but then came back and did an honours degree in virology. Over a part-time period, she also enrolled through the Faculty of Medicine's Tropical Health Program and did a PhD on Burkholderia pseudomallei, the causative agent of human melioidosis. Arla is a research-focused academic, a strong background in industry engagement associated with both animal health and agricultural biotechnologies. She's got a wide range of interests, particularly the application of omics to develop molecular and diagnostic serogenotyping methods to study gene function in relation to virulence and host pathogenicity, to develop new effective vaccines. She's also interested in identifying biomarkers and studying microbiomes associated with improving animal health. Arla has over 100 publications with 3 full pending patents for 2 different anti-tick vaccines and has commercialised diagnostic assays and tools. Her research areas are quite diverse: bovine reproductive diseases, the paralysis tick, the cattle tick, tick-borne diseases in general. She's attained and completed $12 million in competitive grants in the last 10 years, and that has included ARC as well as pharma and industry funding. Together with SCMB Biotechnology Program and SAFs, she's also developed UQ's agricultural biotechnology field of study within the Masters of Biotechnology in 2020. She's got wide international recognition. She's a member of the Bill and Melinda Gates Foundation International Cattle Tick Vaccine Consortium, and she's the specialist tick editor for the International Journal of Parasitology, and she chaired the International Tick and Tick-Borne Pathogen Conference and leads the Australian node of the 1000 Ioxodes Genome Project. So a really broad career that's gone from bacteriology to virology to parasitology to molecular biology to vaccinology. So Arla's done it all. The one thing I'd like to mention, because I've known Arla for a long time, she's a wonderful mentor, both of students and staff, and I think that's really important achievement along with all of her science. So Arla, I'm going to hand over to you to move into the seminar. Thanks, Arla.
Okay, thank you, Pat. There's a few slides I can skip now that you've done a bit of that. So to move straight into it, when I was thinking about what I would like to talk about today, I've done quite a few seminars this year, both at UQ and at different conferences, and I wanted to look at what is it that motivates me and what I do, and it really is sequencing. So at the group currently as it stands up here, I've mentioned that Cumrun and Nomun are primarily supervised by Peter James, but they're all based up on level 3 at Quaffy. So my current interests are commercializing 2 vaccines, looking at biomarkers for various diseases, using various methods, molecular diagnostic methods. Bovine genital compylobacteriosis has been a bacteria that I've been working on since 2003. Looking at the metagenome of cattle associated with infertility in the north, and ectoparasite genomics really to develop vaccines, and would be keen to develop some online genomics tools through some future research. There's an overview of the talk. First, I'm going to jump into this history of sequencing and Sanger sequencing, 1977, PCR developed 1983, and 1990, in the first year of my PhD, I had to sequence to develop an assay for Burkholderia pseudomallei, and this is how we used to have to sequence. This is a very big page gel using sulfur label to get the sequencing data from such a gel, and from that literally sat there with a piece of paper going A, G, C, T, G, C, like, and then use that to develop an assay within my PhD. So then, of course, sequencing became automated. I think we started using this service about 1993. You're all familiar, if you do any Sanger sequencing through this fluorescence detection method, you outsource the running and you receive the chromatograms and you analyse them. So I have to say that since then, the sequence analysis programs that we've used to look at these chromatograms has changed probably every couple of years. I don't know how many different programs we've used. We, for a long time, did our own sequencing reactions and sent them away to the external provider, but now we tend to just send the DNA primers if we do any Sanger sequencing of short sequences. So next, 454, we did a little bit of 454 work, quickly came out Illumina, PacBio long reads, and then Oxford Nanopore long reads. And about 2013, we sequenced 9 Campylobacter genomes using Roche 454 at the end of an ARC linkage project. Using transcriptome data, we also sequenced, we did transcriptome sequencing to identify paralysis tick toxins. We resequenced those same Campylobacter through PhD student Chen Tang Ong, who I'll talk a little bit later, through PacBio and Illumina, and then she resequenced them using Nanopore MinION in-house in the lab. So this is just another diagrammatic representation of where we've gone in sequencing. First generation, the cost was about $3 billion for the human genome. You can see the prices coming down and the time taken down to actually do sequencing. And another way of looking at it, a different diagram showing the short read sequencing and more recently long read sequencing, which is also improving and increasing our ability, less time consuming, I guess, to bring in long sequencing because of the long time it takes to assemble Illumina data in comparison. So Path mentioned a bit about my career path. Yes, started in viruses, and this bacteria was the culprit as my PhD, and then tick-borne diseases and the diseases of venereal disease and ticks, and also a project on screwworm fly. So first project was ELISA development for canine parvovirus, monoclonal antibody production, and then in my honours, I made monoclonal antibodies against an animal ephemerovirus, baromovirus. And then I started working, and the technique at the time for epidemiology of human diseases was ribotyping, and started my PhD with the same group, developing methods for Burkholderia pseudomallei, which causes melioidosis in humans. And then from 1992 to 2010, I was employed in DPI, or QDAF as it currently is. And the text highlighted in purple are areas that I actually took with me along to UQ and still work in, and became part of the 2010 Quaffy Institute as the first 38, I would call us. Sequence data was a very important part of all this research here. And I drew a line here as I did not enjoy doing monoclonal antibody work and ELISA work as much as I enjoyed this part of where I work and where I am now. So the next part I'm going to go into is novel diagnostic and genotyping assays. I want to talk about the tick fever diseases that tick carries in this part of the talk. I'll talk a little bit more about the impacts of the cattle tick a bit later, but generally ticks and the diseases they carry affect 80% of the global cattle population, to a tune of about $20 to $30 billion in losses per year. So this is an old diagram showing the distribution of these tick fever pathogens. Two are Babesia species and one is a intracellular bacteria, showing here like where they appear in the red blood cells. When a gene sustained red blood cell, that's what it looks like. And you can see it's quite a global distribution depending on where the ticks are that transmit those diseases. You can see Australia, it's mainly Northern Australia. That's the same distribution that we have for the cattle tick in Australia. So I always have to put context. Babesia I'm going to focus on now, very closely related to falciparum malaria, which you might have heard of. So Babesia were discovered by Victor Bubbes, and first he called it Haematococcus bovis, obviously thought it was a bacterium, and later named Babesia bovis in his honour. So of Babesia bigemina, there's several tick vectors, as is for Babesia bovis, but in Australia we only have the top one, the Rhipicephalus microplus australis. And so there are outbreaks in New South Wales, because if you remember the map that I showed you, the tick distribution is proper northern, and occasionally animals or ticks sneak across the border, and there are deaths associated with Babesia bovis usually, as it has the highest mortality compared to the other two pathogens. So I have to give you some context about the Australian live tick fever vaccine. It's been made since 1964 at Tick Fever Centre at Biosecurity at TAF. And there are three components to the vaccine, Babesia bigemina strain and Anaplasma centrale to control Anaplasma marginale, and they have never changed. But the Babesia bovis strain has changed regularly over the years because the vaccine seems to break down and they needed to change strains. So what they do is they attenuate these Babesia bovis parasites by passaging them through splenectomized cattle. And their 1964, how to put it, method that they used was, we do 25 passages and it's attenuated, then we use the vaccine. So we brought in, I brought in some genotyping methods to look at all of this, and we genotyped and found that it was already attenuated at 15 passages and it was a more heterogeneous population. And to our credit, that vaccine strain has been used now since 1996. So the publication for developing these genotyping methods is so old that in the journal, that's what it looks like, the black box down the bottom, but I have found some old gels. What it's showing is the 25 passage strain as a strong population here, and then 15 passages, there's 2. So it's more heterogeneous. It's another way of showing it, 12 passages, 2, 25 passages, 1. So occasionally, though, things happen. So it was, unfortunately, Australia was responsible for transmitting Babesia bovis to New Caledonia in recently vaccinated cattle. They're supposed to wait 42 days until the parasites go down to a carrier status level, but they were sent overseas. And it was that genotyping test that confirmed that the cases in New Caledonia was the vaccine strain, which had reverted to virulence. So it's quite odd, I didn't know what was going on. I get an email from Tick Fever Centre going, what do you think of this gel? And I go, what's in it? And then we worked out what happened. So we decided to upgrade that method, because I don't know if you noted in those gels, those fragments were 6,000, 3,000 base pairs, quite a difficult PCR to do for a diagnostic lab like Tick Fever Centre. So we developed this assay with Jess Morgan, which uses a tag, and then you run it on a sequencing gel and you get peaks of different sizes of genes. So we decided to look at the internal transcript spacer gene, which is often used in parasites for strain differentiation, as opposed to the 18S or the large subunit RNA sequences, which are more conserved and better used for phylogeny. So we cloned and sequenced a whole lot and clustered them and decided that we could go on a size assay. And this is the result. This is the old assay showing you the bands we used to get from different isolates. And then over here, these are the different peaks that we get. So we get much more data. And as you can see, the normal vaccine strain has got these 2 populations, but after it's gone through a tick and reverted to virulence, it has another 2 populations. So if you look at these field isolates, this one probably wasn't caused by the vaccine. This one perhaps. This one definitely matches this up here, which is the Dixie through ticks. So it's likely to be actually caused by the vaccine strain in an animal that probably wasn't vaccinated. So we updated it, and this has been published in 2016 by Bing Zhang. We also did some PCR development here as well, but the focus was the genotyping assay. So although this isn't agriculture, it just shows you how things lead to other side projects. And we did some studies to identify a new babesia species in the eastern grey kangaroos. And we did this by 18S sequencing. And technically, obviously, this is a new species. So kangaroos do get babesia infections, and they appear to happen when they're in captivity. So now there's a species there. So just a moment to acknowledge Tick Fever Centre staff. There's so many, I don't even know if I've missed any. The 4 are still with, that I've highlighted here, are still with DAF in different positions, and they primarily distribute that live vaccine that I talked about. But they also distribute this tricampus sampling tool, which takes me to my next topic, which is bovine infertility or venereal diseases. We actually developed this tool, and it was trademarked while I was in DAF. And the irony is that now I have to pay a lot of money to buy it to use it in my work. But here is Jeffrey Fordyce, who did the work, and we demonstrated that you could isolate more organisms that cause venereal disease using this tool. So over to bovine venereal diseases. Some of you may be uncomfortable, but don't be. There are 2 diseases, again, like tick fever, a bacteria and a protozoan. So over here we have Campylobacter, the gold standard diagnosis at the time, and still is actually, culture and biochemical testing to differentiate it from a subspecies that does not cause venereal disease. And over here is Trichomonas, caused by Trichomonas fetus, and the gold standard is diagnostic culture and microscopy, but our assay that we developed has now been used as a diagnostic standard. There's a bit of a life cycle here of Trichomonas that I won't go into, but obviously the diseases are passed during mating. Bulls are asymptomatic. They don't cause any problems, but causes infertility in female cattle after mating. And here are just some morphological pictures of Trichomonas fetus. So bovine genital campylobacteriosis is quite a worldwide distribution. I suspect there are more cases in South Africa because there are publications that come out of there. There is a vaccine that we can use in Australia that was developed in the 70s that works quite well. But for Trichomonas or trichomoniasis, there is a similar distribution, but there is no vaccine available in Australia at the moment. So again, I like the context. Trichomonas sit here in the protozoa over in this side, near other human pathogens, except it's a venereal disease. It's also a venereal disease of humans called Trichomonas vaginalis. And again, a tree, which I like, and Campylobacter fetus, subspecies venerealis, sits in here, closely related to fetus, and several other organisms around, like Hyointestinalis, now Campylobacter urolithicus, Archobacter cryoaerophilus, and Campylobacter spitorum, cause us diagnostic issues in our development. So at DPI, I heard this story that venereal diseases were diagnosed using microscopy and using culture. And I knew enough about bacteriology or microbiology that's impossible to grow Campylobacter. It's just impossible, very difficult organism to grow. So I thought, really, is this how they're doing things in DAF? So we decided to do something about it. We thought we could look at the sequences and develop some qPCR real-time assays, which were the rage in the early 2000s in changing our diagnostic methods. And just to be authentic, that was the MLA logo of that time, which led obviously to an MLA project. And for Trichomonas fetus, we looked at the internal transcript spacer as a region for an assay. And for Campylobacter fetus venerealis, there was a PCR assay that was in existence. And this band indicates it's C. fetus. If it has this band, it indicates it's C. fetus venerealis. So we took this band. No one knew what it was. We sequenced it and designed a real-time assay for it. So happy days. Over here on the side gives you a diagrammatic representation of what qPCR probes and how they work. Essentially, if the probe binds the tag nuclease, comes along and releases it, and we get fluorescent signal. And so we published the real-time assay for Trichomonas fetus, happy days. And we also published an assay for Campylobacter fetus venerealis. We thought we'd achieved everything that we had to. This assay is done very well. It's commercialized by Thermo Fisher. And this one, no, done badly. So there's some cross reactions happening with other organisms, some of the ones I talked about before. and this was determined by 2011. So success, fail, but we kept going. So you can see there's a couple of years have passed before we got an ARC linkage off the ground to focus on genital compylobacteriosis. And in this project, we, oops, sorry, too fast. In this project, we screened bull samples, about 1,000, using several different qPCR tests that were available at the time. Now we know they're not specific, but it was a tool to find samples from bulls that were potentially positive that we took back to the lab, and then we tried to grow in PCR. So we identified in this niche several other species that are normally not in the venereal niche that I'm talking about. They might be fecal, they might have other distributions. So what I'm trying to say is if we use lab strains of these, all our assays are specific, but for whatever reason, they also coexist in the bull prepuce. So we got some data from an Argentinian strain. We looked at, we did some 455 sequencing that I mentioned before. We published the Australian strains of C. fetus venerealis genomes, and we also decided at the end of the project, as you can see after, through an honours student, we sequenced a conserved gene, a chaperonin 60 or a heat shock protein 60, to confirm the qPCR and the microbiology identifications, which did not match. They didn't agree with each other, but neither method was 100%. And it was only sequencing, just normal Sanger sequence of this gene, that enabled us to identify what these species were. So there's a diagrammatic representation. This is what Campylobacter looks like on a plate. It's in tiny little colonies. This is what happens in a diagnostic lab when they try to grow it as from the transport medium. It's overgrown by Staphylococcus or other Pseudomonas organisms. So you can see impossible to isolate. So essentially, we had these sort of results that didn't really match, didn't really tell us exactly until we did that sequencing. And from 1,000 samples, we had 16 true positives of fetus venerealis from this big study. That was the Campylobacter, so ARC linkage team. We can see some common faces. Mike McGowan still worked with him, and also with Gree, and Bromman was involved on this project. And just moving right along, Yusra was an honours student that did the sequencing of those housekeeping genes. This solved a lot of problems in a very short project. So move forward again, probably another 4 years before we had a project up and running. So we knew that housekeeping gene that we used, we knew there was one SNP difference between C. fetus and C. fetus venerealis. So we developed a TaqMan SNP assay, and we have 2 probes in the assay, same primers, except there's 2 probes, one labelled with FAM and one labelled with VIC, that can differentiate which species it is, and it doesn't seem to amplify other species except at high CT scores. Those of you that know qPCR, the higher the score, the less sensitive it is. So a true positive is around in the 20s or below, but once it gets into the 30s, we're not sure. Regardless, DAF Biosecurity have tested this assay and they're quite happy with it. They use different equipment to us and modified the conditions, and we're still working with them at how to move forward, as we've had a lot of requests for this diagnostic assay. So bring along those same genomes that I talked about before. Chen Teng-Ong, PhD student, come along, and she finished those genomes, and I got here PacBio and or Nanopore in short reads. What I'm trying to say is we send them away for PacBio and we couldn't get a single contig or assembly. So you can see some of them still here. This is not an updated table. She has updated this and I believe has single contigs for all of them, but she managed to get the single contigs by redoing them herself on Nanopore. So I have to thank Ben Hayes' team. They were using the MinION next to us, and Chen looked over the shoulder and repeated them, and we got better results. And I'm not saying that PacBio is bad, but I think when sending samples away for sequencing to a sequencer provider can be a very generic process, whereas if you have it in-house, you can optimize it a bit better and get it to work for you. So continuing on with Chen's work, she looked at those isolates, if you recall, Hyointestinalis, the Urolyticus and the Spatorum that I talked about before, compared the genomes. is in the heat map here, as you can see. So the C fetus, fetus, C venerealis, very similar with each other. And there are some similarities across, and also looking at the C fetus venerealis that she assembled together, she was able to show that all of the previous diagnostic targets that we were using and everyone in the world were using were in these variable regions. This is the PAR-A, that assay that failed, that we found cross-reactivity with other organisms, which we are famous for publishing a failed assay, but it happens. And so this work has shown us what happened before when we didn't have the tools to do that work before, didn't have genomes when we developed the diagnostic test. It's Chen. And just to summarise all of that about Campylobacter fetus venerealis, because it's quite long, started a long time ago, we sequenced this PAR-A gene, and TaqMan assay ended up getting non-specific positives. A lot of this was by work of Leah Ingen, who finished her PhD around about 2013, I think. And separately to us, the discovery of the pathogenicity islands in the genomes of these species was published in 2010. So we obviously found that the main differences between fetus, fetus and fetus venerealis appears to be virulence genes, which do not appear to be the best genes to use for diagnostics. So Leah, also with DAF biosecurity microbiology staff, were trying to identify, trying to identify the isolates, Leah using qPCR and the other lab using microbiology, and their assessment did not match. We did some Illumina sequences, I mentioned, of C. fetus venerealis, and then Yuzra came along and did this sequencing to confirm the identities that could not be confirmed using the previous qPCR methods and standard culture. So now step forward to Chen doing her PhD. She resequenced them all using Illumina and PacBio, resequenced the long reads using Nanopore to get a single contig, and identified another SNP that we could use to develop another assay. And Priya Pius is with us this year doing an extensive biotechnology master's research program, and she's been evaluating the new test, which were also recently sent to DAP. So we've sort of got test one and another test, see how we go. So biomarkers for disease resistance. So looking up definitions of disease biomarkers, there is plenty of research in the human space, and they're really broad definitions. So I'll go to the broadest one on the slide, which is any substance, structure or process that can be measured in the body or its products and influence or predict the incidence of outcome or disease. So I found this really cool diagram that really helps me with the story. You can see there's lots of sources of samples that can be used for biomarker discovery, lots of different molecules that can be used, and lots of classes. There's all sorts of types of biomarkers that are used. So in our work, we can add here skin. We use skin biopsies in cattle, and what we've focused on is proteomics, and we've also focused on RNA-seq microRNA sequencing as shown here. So that's where we have focused our work on biomarkers. So I had to explain a little bit about the host for the cattle tick. Now, I'm not going to go through this diagram. Those of you that have forgotten your Roman numerals, there's a bit of a legend on the side, but essentially cattle move around the world with the migration around the world. Now, cattle ticks evolved on Bos indicus or Brahman cattle. It's shown here with the hump. India, obviously, the name. And they're naturally tick resistant. The ticks evolved on these breeds, and they were imported into Australia up into Darwin around about 1872 and brought with them ticks. And then earlier, as you would know, Holstein Friesian or Jersey cattle came with the Europeans to temperate climates. They are very susceptible to ticks, and hence when the populations merged, we've got a lot of sick Bos taurus animals that cannot handle the ticks. But what this gives us is host differential, where we could compare the immune response or the genetics, anything to try and work out how to improve breeds. So I'm going back to the beef CRC where we looked at immune markers. We looked at these two breeds, literally, they're the ones at the Jarrah Hills that we used. and we did comparative studies between them. I'm showing here Emily Piper, who we call Emily number 1. That will become obvious as the talk goes along. She published about 4 papers about these studies using microarrays, immune markers, can't remember, published 2008 to 2010 as part of her PhD. But as a team, we were a bit concerned that perhaps the differences between these extremes were too much and too much genetically linked. So we decided to look at crossbreeds. So there are many crossbreeds. Here on your left is a shorthorn, and on your right is a Brahmin. And what they created was Santa Gertrudis. So they're effectively about 5/8 Bos taurus and about 3/8 Bos indicus, and they have quite a good resistance to ticks. And these are the Santa Gertrudis that we did studies on. As you can see, these were completed in 2009 and published not so long ago by Emily Piper, Emily number 1. And these are essentially people that were working together in the big CRC team. And we also did a lot of immunohistochemistry through Con, and he found some possible markers, which I'm not going to talk today about. And we actually used the serum from these trials to get our proteomics working through Ali and on our current MDC project. So we still had, he was able to get his proteomics working and have a look at the serum proteome to correlate to this previous work that was done by others so long ago. So that brings me into this project called Biomarkers and Accurate Phenotyping for Selecting Resistance to Cattle Ticks, Buffalo Flies and Buffalo Fly Lesions, which we lead together with Peter James. So with cattle tick, essentially sort of picked up where I left off and using more recent technologies. And with buffalo fly, starting sort of a little bit further back to cattle tick, and these are the PhD projects of Noman and Kamran. So we decided to focus on Brengus, which is Angus, Bos taurus, crossed with Brahmin. So they're quite popular in Northern Australia. So there was a Santa Gertrudis study. We essentially repeated it in Brengus cattle. Just going to see how I'm going for time. Better hurry up. So we did the same phenotyping that was done for the Santa Gertrudis cattle. We infested them weekly for 13 weeks, then separated them on tick scores. You can see here, lots of ticks, less ticks, and these extremes were used in the subsequent experiments. So I can't look at Emily's notes while I'm in presentation, no, but she has looked at RNA-seq and small RNA, and she's looked at pre and post tick infestation, and she's also looked at the difference between high and low host resistance. And you can see differentially expressed genes between high and low host resistance steers. There's also commonly that gene expression does change on either breeds with tick infestation, noting that there are tick-naive cattle when we get them. So to follow up here again, she has done some time port contrasts as well, and she's found gene expression changes linked to several biological pathways, and hopefully will improve the understanding of host resistance to tick infestation. So this was a comment Emily put on one of her slides, which I didn't use as an intro slide. She said, and it was very well written, host resistance to ticks is a complex trait that's difficult to measure. So using RNA-seq, the study contributes to identification of candidate genes associated with host resistance that can be used in biomarker-assisted selection to control tick burden and improve cattle welfare. Thanks, Emily. So next over the proteomics, which is Ali. He used the same animals. He's presented it differently diagrammatically. He used the blood samples, and he's decided to do swap analysis, which is the proteomics version of statistical, trying to explain it. Using several samples, you're able to have a more quantitative comparison of your samples. If anyone has any questions, ask Ali. And he's able to quantify and find proteins differentially insusceptible and resistant through the same sort of time course and his study that Emily did, except using proteomics. So he did a string analysis, and why we're interested in looking at naive cattle is if we want a biomarker, we want it to work in cattle unexposed to ticks as well as cattle exposed to ticks. So he found a high number of differentially expressed proteins in the tick resistance before tick exposure, and he found several involved with immune response, lipid metabolism that he's highlighted here, which is very interesting. And the important part, it confounded what he'd previously found in his Santa Gertrudis study, which was his warm-up for this particular project. So again, potential biomarker for tick resistance. So I haven't given you any background about buffalo flies, so quickly I'm also conscious of time. Buffalo flies cost about 98 million and affect cattle weight gain and dairy cow milk production. And it's generally in the north of Australia, similar distribution to the cattle tick. So Kamran and his PhD is also looking at the serum proteome in ticks, in cattle with high numbers of buffalo fly and low. And he's still in the process of going through this. And he has shown that most of the proteins that he found were associated with immune response, suggesting a strong immune response to fly infestation. And there was the development of strong immune response may contribute to the low fly burden on the resistant cattle. And he showed that in those with the lower fly burden that there was not many proteins involved. And we haven't gone into the details of these, but again, this is important in buffalo fly host resistance and biomarker development for this project. So I mentioned fertility before. We're also looking at biomarkers for bovine genital campylobacteriosis. One thing that has been bothering me or others for a while is often cattle that are vaccinated will still come up positive in qPCR diagnostic assays. So we thought, is there a biomarker that we could develop that tells us that this animal is immune? It might harbour the organism. So this is part of Sogra's PhD. She's hit the road running. She only arrived in April this year, and see how we go. So I'm meant to move along. Microbiome research. So we're looking at the reproductive microbiome. Just a little bit of background. You might have obviously heard about microbiome studies in human using 16 RNA sequencing, and there's total microbiome DNA sequencing. So 16S is very fast, but there's often a real challenge that there's a lot of host material before you can get down to the microbiome or the bacteria. So again, Chen Tang-on is back again, part of her PhD. So using traditional methods, she had something like 99% host contamination. And she has written a paper where she's evaluated the host depletion extraction methods. This paper is submitted under review. We're still waiting to hear from it. This is this other MLA donor company project. She did some spike samples so she could see what she gets, puts in, what she comes out, which was useful. And she has done, she has did a whole lot of methods, which I'm not going to go into detail, but I believe this one down here gives the best yield. So this paper we're waiting to hear on. In the meanwhile, she's modified her sequencing and she's using Nanopore. Why? Because she can actually set it up so that the bovine host DNA doesn't get sequenced. So this is called adaptive sequencing, so very useful. You can see over here in green, Nanopore adaptive, yay, really good yields. Nanopore normal without using adaptive, great. Using the methods that she had in the previous slide, but then changing the game in the way she does the sequencing. And also it gives a better, higher percentage of taxonomical hits, and she can get a higher percentage of functional hits and just a lot more information. She's in the process of analysing this data. So where would this lead? I mean, if we identify a healthy microbiome and we can be indicative for superior fertility, maybe we can develop a prebiotic treatment for cows before mating that will have the healthy microbiome and be healthy fertility-wise, ultimately improving the fertility in the Northern Australian beef industries. So quickly, very, very quickly, so I'm looking at the time. So I talked about tick fever. Now, in the terms of impact of disease, tick fever sits way down here. The cattle tick is way up here, causing the largest amount of losses. Buffalo flies, spoke about before, also high. Hence why, I'll say 16 years ago, my focus went to ticks. So what are they? They're mini vampires. They feed on mammals, birds, reptiles, and amphibians. They're second to mosquitoes as the vectors of some really important pathogens all around the world. There's about 892 species worldwide. There's specific hosts on different continents. There's about 70 species in Australia. 5 were introduced. And this is the cattle tick. It was an introduced species, as I mentioned, from the north, from Indonesia on Bos indicus cattle. That's how bad an infestation can look like on a susceptible breed, looking like an Angus there. And then, although this is not agriculture, we went to paralysis tick, which those of you that have dogs and cats know a lot about this yourself as you treat them. Paralysis tick also causes outbreaks and deaths in cattle in northern New South Wales in areas where they don't treat for cattle tick because it's not there. So there are still outbreaks which cause losses in cattle. So similar slide that I showed you before, the life cycle of a cattle tick. It's a one-host species. It prefers just to develop on cattle. It's a 21-day life cycle mostly, and as I always say, these ticks are not to scale. So we get larvae that climb up, feed nymphs, feed into adults, females, males mate, females grow quite large, drop to the ground, lay eggs, and the cycle continues. Now, what we have a challenge in phylogeny world, in the parasitology world, is that in Australia, this particular species has been renamed Rivisicolus australis. Now, to me, this is a bit odd. It's introduced into Australia, but that's the taxonomy. That's what we're with now. So you'll see us interchanging Microplus and Australis because Australis sort of was published in 2014. So this is a worldwide distribution of the cattle tick in black, and the stripes done very unprofessionally by myself on this map is where this tick has spread since the publication that I can tell here. One of the main issues was that the pesticide resistance of ticks is globally increasing, and in some countries, they don't actually have any chemicals they can use. Ticks are resistant at all. So moving very quickly, because I'm conscious of time, cattle tick genome is huge, but it has a 70 to 85% non-coding region, which makes assembly very challenging. So I'm not going to go through, this is a summary of the technologies used to sequence different tick genomes. And you can see as you go through that the Illumina methods keep improving, the long read methods keep improving, but we still don't have a chromosome assembly for any tick species at this point. BM86 was developed by CSIRO in 1986. It was a gut protein found to work quite well as a vaccine here. It wasn't adopted by the beef industry, so it was discontinued in 2010, largely because it needed several boosts a year to be effective. And as you know or don't know, in the northern beef industry, cattle are only mustered once a year. The Cuban vaccine still remains in use. Cuba is a small island. You can go around and vaccinate everything quite easily as many times as you want. So it's been quite successful. There's also some sequence variation around the world, which led to our vaccine program, which is called a reverse vaccinology program. So reverse vaccinology essentially is a way of looking at the genome or the sequences that you have and mining them with computer predictions to find possible candidates, test them in an in vitro model, and then test them as vaccines. So when we started this, there was no genome. All we had was an express sequence tag library of about 13,000 sequences, which we used to do this work. Now, this is our pipeline. I'm not going to talk about today. It's a seminar in itself. We did some discovery. We did a lot of microarrays. We did a lot of QRT-PCRs. We did a lot of predicting. We predicted epitopes in the sequences. We fed ticks in the lab with antibodies that we'd made to particular candidates. Then we tested them in mixtures, and then that was where we got to at the end of the beef CRC, and then through MLA funding, we've continued to screen, shown here. So these are the main essential attributes, which I won't go through because of time. Essentially, we picked transcripts that were recognised by tick-resistant cattle, that bound B-cell or antibody binding epitopes, what worked well in the in vitro model, and what worked in vivo. Sorry, it's my timer. So moving along, this is where we're at now. Our most recent trial was a longevity trial. We only gave 2 boosts compared to our normal 3 boosts of vaccine, and we did 2 challenges about 6 months apart. And this mixture of 2 proteins, which everything here is top secret, as you can see, this was a great result because the efficacy went up after the second infestation. And efficacy going up is not something that we've seen before, and we're very excited. Happy dance. This is where we're at now. Those ticks from that happy dance, the ticks are black and they don't lay eggs. This is what a normal tick laying eggs looks like. And Nimita Ramachandran in the lab is doing a master's extensive, and she's looking at the transcript sequences from these ticks and comparing to see what happened in those, which is interesting. We're using Nanopore sequencing, and what is great is you get the whole transcript coming from the Nanopore system, and there's no need for Illumina short read polishing. But this is really exciting as far as Nanopore goes, and looking forward to seeing this data. So just going to touch on commercialization, where it's at. 2 very large grants, then MLA funding, and then we spoke to a bunch of companies, and then we had an MLA donor company project with Elanco. Now, as you know, companies keep changing. They acquired Bayer Animal Health and changed their focus to parasitic drugs and not vaccines. So we're finalising our P assignments, and we're going to speak to new companies about moving this vaccine forward. So quickly across to paralysis tick, don't have time to describe it, but any of you that have pets are familiar with these drugs. So, and if your dog happens to end up in a vet surgery with paralysis, they're giving this tick antiserum, which is produced from tick hyperimmune dogs, can still be fatal, and there are adverse reactions to this, and some animals still require euthanasia. So we did transcriptome sequencing of collected with ticks that we collected from paralysed dogs and cats from local veterinary surgeries. We dissected out the guts and the salivary glands and essentially sent away for Illumina High-Seq. We identified, there was one toxin identified in the 90s. We identified homologs to it, picked the most immunogenic, and did a proof of concept dog trial. So this is just a tree showing you all the different toxins. The ones that have got seq next to them were discovered by Zoetis, but were never tested. The HTs are our names for them. They're very short peptides. So we did our trials with peptides. So we did a proof of concept dog trial with 8 HTs, and they were protected from challenge and paralysis, and unvaccinated dogs required treatment. This paper has been submitted finally after review through UniQuest and also Elanco, which took some time, but this project is finally being published. So again, we did some preliminary work before we obtained the grant, and it was an ARC linkage. We started in 2013, and talking about companies, Elanco bought Novartis, and the Elanco leaders were made redundant. We had new leaders on our project. Proof of concept dog trial. Then 2015, Elanco withdrew interest because it's not a global problem. It's only the east coast of Australia. UniQuest has helped us to get to a patent position, and we had lots of meetings with companies, other companies not interested, and we actually also did a proof of concept use expression of these toxins, which is useful for commercialization rather than using synthetic peptides. So we're now with this small company in Australia who are going to change their business. Hopefully moving forward, they'll be pulling out this vaccine. So a lot of people on those 2 projects, they started a long time ago. I'm not going to go through them. I'm just going to highlight that aside myself, Manuel, Matt and Felix from the USDA are the inventors of the cattle tick vaccine, and Manuel and I, did it again on the paralysis tick vaccine. Really thank a lot of people, it's been a lot of work. Moving on to the final part, give some time for questions. I'm going to flash through. Kamran has done some Sanger sequencing of he's done some proteomics to identify these proteins, which are found to be significant in the hornfly, which is the northern hemisphere. It's a subspecies of the same species, except it's Hematoba irritans. So he specifically went looking for these proteins. And he was able to find several peptides that, at least for these 2, he was able to find peptides, and in PCR he's able to find more, which is good. What we decided to do was do a transcriptomic analysis of the internal buffalo organs. This was through a master's in bioinformatics student who just finished. She found 37 transcripts of antigen 5, 16 of hematoba, and 9 for thrombostasis. What we still have yet to do is to convert her assemblies into a database so that they can use the buffalo-fly proteomics searching, and also look at her data for more immunogenic proteins, possibly future vaccine development. She did very well. Ali has been looking at sequencing the buffalo genome, again with Nanopore and with all of the work they've been doing in the lab. It will complement everything that we've done so far in proteomics and transcriptomics. Noman has been looking at Stefanophleria, which is a nematode found to be in buffalo fly lesions. Running out of time, there was no sequence data. With Jess's help, we did some sequencing. Jess Morgan developed a PCR assay, and he's found that the nematode exists in the lesions in the north of Australia, but not in the south, which is interesting, yet there are lesions in the south. He's also looked at some bacteria. He's done some nanopore sequencing to look at the isolates of Staphylococcus hyacus that he's found, and he assembled data, and again, Nanopore comes to the rescue. Also looking at sequencing the Trichomonas genome and developing a whole killed protozoan vaccine for Australia, and really want to assemble one tick genome, that would be great. Looking at poor C instead of high C in the Nanopore, how to put it? I better stop. Okay, so these are acknowledgements. You can see there's a bunch of people that are in both places. It was Chen who was looking over Ben's staff, Lone, looking at her Nanopore, and she, so I really thank Ben's people for their help here. And James Cook, Con Constantino was in the Beef CRC, continues to work with us, as does Nick Johnson, who was involved in Beef CRC work and the Santa Gertrudis trial. And these two ladies here, who Jess Morgan and Bronwyn Venus, who were both in DAF and in coffee. Jess chose to go back to DAF after 5 years in coffee, which was heavily involved in a lot of different pieces through there. It was worth mentioning. And that's it. Thank you for your time.
Great. Thank you very much, Arla, for a wonderful overview.
Sorry about going over time.
There are some questions, but unfortunately, I think we're pushed hard against the closing time. So can I encourage people to directly contact Arla if they've got their questions or want their questions answered? I'm sure Arla would be more than happy to do that. So apologies for the inability to answer questions, but we heard a lot of wonderful science, Arla. So we're
I hope I've convinced everything that sequencing gives you solutions. So.
Sequencing is the answer. I've got that written down here. Okay, so we'll have to close that particular or Arla's talk today.
You've got 2 minutes.
Yes, but we've got to advertise next week. So Arla, if you could change the slide.
I'm trying to.
Our seminar next week.
Oops.
That's it. Okay, our seminar next week is entitled "Investigating Threats to Our Green Gold: Blight and Dieback of Avocado," and it's going to be a multi-author presentation. Associate Professor Elizabeth Dan, Dr. Akila Prabhakaran, my apologies if I've got the pronunciation wrong, and Montana Hickey from Coffee Centre for Agricultural Science. Don't forget to visit the seminar's webpage for more information. I would like to, Arla, thank you very much. That was a wonderful seminar. It shows the achievements of someone who's dedicated themselves to a lot of work across a lot of diverse fields and also very applied research at the same time, both scientifically great, but also applied and great relevance. So
is there another slide?
So thank you to our audience for attending today, much appreciated. If you enjoyed the seminar today, please do the poll at the end. And if you haven't already signed up for our seminar invitation list, please do so at that coffee.uq.edu.au/science seminars. With that, unfortunately, we'll have to now close and let people move on to their next activities. Thanks again, Arla. Well done. Thank you.
Thanks for listening, everyone.